here. Here we go. Okay, lovely. So we're back for our last talk before the break. So it's my pleasure to introduce Marieke Mur from the University of Western Ontario. Dr. Mur is an assistant professor in psychology and computer science and a core member of the Brain and Mind Institute at Western University, where her research brings together psychology, neuroscience, and artificial intelligence to understand how the human brain supports perception and cognition. Dr. Mur performed her PhD in visual neuroscience with Nicolaus Kriegerkort, I'm sorry if I mispronounced that, at the Maastricht University in the Netherlands, as well as the National Institute of Health. She then went on to pursue postdoctoral research with John Duncan at the University of Cambridge in the UK. And in her current work, she combines neuroimaging with computational modeling to understand how the human brain transforms sensory signals from the outside visual world into meaningful representations that support cognition and action. So her talk today is entitled Representational Transformations in the Human Visual System. And without further ado, the floor is yours, Marieke.
All right, thank you for this nice introduction. Thank you for inviting me. So I'm excited to share my research with you. So I'm just going to share my screen. Let's see, over here, share, go here. So I assume you can see my slides now, right?
Yep, and
okay.
We can see your mouse as well.
Okay, perfect. All right, so let's get started. So one of the major questions that my lab aims to address is how does the brain transform images into meaning? So while I want to focus on the transformation aspect here, I do want to first provide some background so that we're all on the same page. So the model or the system that we're trying to model is the ventral visual system. So this is a system that we know is involved in object recognition. And we want to understand how light that hits the retina gets transformed into a higher level representation, a meaningful representation of the visual environment at the level of IT, or inferior temporal cortex, which is shown here in gray. So let's first look at that and ask what type of information is represented about images in IT. So in order to do that, what we did is we presented visual images to human participants, in our case, while they were laying in the fMRI scanner, and we measured their brain activity in IT. And here we see the images. So you can see that they're from a range of categories, including humans, so that's faces and bodies, animals, also faces and bodies, then natural objects and man-made objects. So now what we did is we developed representational similarity analysis to characterize the information represented about these objects by the brain. And what we do there is for each pair of images, we can estimate response patterns in the brain. So in our case, those were response patterns across voxels. And then what we can do is we can compute the dissimilarity between these response patterns and then store that in a matrix. So this is the representational dissimilarity matrix, or RDM, which contains for each possible pair of images their dissimilarity in the response patterns elicited by these images. And you can see the stimuli are on the axes. So I'm just going to show that here. They're ordered according to category. And then what we do also is that we color code these dissimilarities so that blue colors mean that two images elicit similar response patterns, and warmer colors indicate dissimilar response patterns. Now, this matrix will be symmetric about a diagonal of zeros. And then for these two images here, which are faces, we would expect similar responses. So they would yield a blue color. And then if we replace one face with a banana, we would expect to see more different responses. So now I'm going to show you actual data from these human participants for IT. And the data were averaged across the participants at the level of the RDMs. And when we look at this, we see quite clearly that there is structure in there. So we can see that there is this animate-inanimate division, where you see clearly that if you have two objects where one is an animate object and the other is an inanimate object, you see that they elicit different response patterns. We also see a quite clear cluster of human faces. So they elicit similar response patterns in IT. Now we can show the same information using multidimensional scaling in a lower dimensional embedding space. So here, the distances between the images reflect the dissimilarity in their response patterns. And that means that images that are placed further apart elicited dissimilar response patterns. And this is a different way of visualizing the data with the least possible distortion. And what we see here is that if we color code these images, according to their category membership, we can again see this same information, that there are these clusters of animate and inanimate objects and also human faces. So I'm just showing this because I'll use more of these multidimensional or MDS plots throughout the presentation. What is quite nice now is that we cannot just use this method to characterize what type of information is represented about the object. So this is the meaning, right? Where we can see this is really ecologically valid or ecologically relevant categories that we see in the representation. We can also use the same approach and compute object representations in models. So these could be deep convolutional neural networks, which will feature later in the talk. And there we can just have response patterns across model units in response to these images. We can compute an RDM as well there. And now at the level of the RDMs, we can ask how well the models, we may have different versions, right, explain the brain data. So,
Marieke, sorry
yes.
to interrupt. Would it be possible to click on the hide button? There's like a little banner.
Oh yeah, of course, here. Yeah.
Thank you. We couldn't see the bananas. So.
All right. Well, there it is. Okay, so this is background, right? So now we have this background. Now let's move to the transformations. So how does the brain transform images into meaning? We've looked at what the meaning may be, right? These high-level representations. But now the question is, what about the transformation? And let's make that first a simpler problem just to get started and say, what about we start with the early visual representation? So that would be in V1 and 2. And we want to predict now, maybe not the representation across the entirety of IT, but let's just first focus on category selective regions, which are part of IT. For example, in this case, I'm going to look at PPA, which is the parahippocampal place area, responds as a whole more strongly to images of places than to other object images. And now the question that I'm going to address is whether I can predict the level of activation of PPA from the early visual representation. So let's first look at the early visual representation. This is going to be an MDS plot again. And here I've color-coded the images by category. So what we see here is that we already see some clustering of places in blue and faces in red. So now the question is, can we actually predict from this representation how strongly PPA is going to respond to these images? So if we could linearly predict the activation level of PPA, then we should be able to place a ramp over this space that predicts the PPA activation level. And I'm going to now add the PPA activation level so that we can visually inspect whether this ramp would actually explain that level of activation. And I'll do that by adding a third dimension. So this is just the two MDS dimensions in early visual cortex. But now we can add the PPA activation as a third dimension here. So that will be the height of the bars. So when we do that, we indeed see that, as expected, PPA responds more strongly to places than to other object images. And we also see that if we place a ramp over this space, it wouldn't be able to fully explain the height of the bars because there's this drop in activation when we move from the places to the other object images. So this suggests that it's not enough to do a linear readout, but we also want to quantitatively test this because this is displaying the early visual representation in a lower dimensional space. So we want to incorporate or analyze the entire high-level representation. And we do that simply by using regression. So what we do here is we use the voxels in early visual cortex and linearly combine their responses to best explain the activation level of PPA. So this we refer to as functional readout analysis. And what we see there is if we do this, so I won't go into the details here, but if we do this, we do see that this does explain a significant amount of variance in PPA responses, but we do better if we add a nonlinear model component. So that's consistent with what we observed in the visualizations. So this suggests that our model is too simple, and that's also consistent with previous literature. But now what we can do is to extend this model, make it more complex. We can do this in several ways. So we can start by saying that PPA actually consists of multiple voxels as well. It's a pattern of response. We can also add intermediate layers of or stages of processing. So that would be, for example, V4. Then add nonlinear activation functions to make the network more flexible so that it can learn more complex transformations. And then we also can say, well, we want to go all the way from the image, which is our original goal, right? And end up with an output, which could be a category label like house. So once we've done all these extensions, made these extensions, now we've arrived at a deep neural network. So deep neural networks have become, as we all know, quite popular over the last decade as models of the ventral visual system. And there's multiple reasons for this. So they're image computable, they're computationally explicit. Of course, importantly, they are also reaching and even surpassing human level performance at standard object recognition tasks. So while these developments of these networks were mostly driven by computer vision research, then of course neuroscientists became interested in these networks as well and started asking, can we use them? Are they good models of the ventral visual system? So that naturally led to the question, how well do these deep neural networks predict brain responses? And I'm just going to show you a figure from a recent paper that summarizes quite well what's been going on in the literature or the findings in the literature over the last decade or so. And this is for human data, but similar findings have been shown in the non-human primate literature. So what we see here on the y-axis is the correlation between the RDM that's computed for IT that we saw earlier in the presentation, and then the RDMs that are computed based on representations in the models. So in this case, what we see, these bars here are an average across multiple feed-forward models, including AlexNet, VGG, and ResNet. And the first point to notice or to explain here is the noise ceiling. So this indicates the estimated performance of the true unknown model given the noise in the data. And that's where we want our models to be. So now what we can see is that we see different bars for slightly different types of models in terms of how they were trained, so whether they were trained or not on object categorization, and whether they were fitted or not to best explain brain responses. We see that training plus fitting yields the best results, but there's two main points to make here for all these different versions. And the first one is that they do explain significant amounts of variance in the brain responses. So these deep convolutional neural networks are definitely promising models of the ventral visual system. However, we also see they do not reach the noise ceiling. So that means that they leave significant amounts of variance in brain responses unexplained. And that's an important point because that's where we want to make improvements. And that yields the question, what is missing from these models? So in order to address this question, what we did in recent work in the lab is we started to look at representational dynamics. So it's important to know or to emphasize, of course, we all know this, but the representations are dynamic, so they unfold over time after stimulus onset. And in order to capture those representational dynamics, what we did is instead of analyzing fMRI data, we analyzed source reconstructed MEG data. And what we see here, so this was work done in collaboration with Radek Kitri and also Tim Kiesman. What we see here now is that we have RDMs for different stages of visual processing across the ventral stream. So that's, as before, we also had this for fMRI data. But in addition, and I'll show this in a moment, these are movies. So the representation, we can capture how it evolves over time at a high temporal resolution. And these RDMs, they're based on the same images as before. They also have the same organization on the axes of the stimuli. And what we can see here is the color code is slightly different, but blue again means that two images elicit similar response patterns, and yellow-green means they elicit different response patterns. So I'm just going to play these movies, and we're going to look at them, and I'm going to comment on a few things that happen. And here in the right bottom corner, we see time after stimulus onset. So the images were shown for 500 milliseconds. So once we start playing, I hope this works well. What we see initially, there is no structure, no information, because it takes time for the information to travel to V1. And then what we see around 50 milliseconds after stimulus onset, we start to see that there is this cluster of human faces, which means that they start to elicit similar response patterns. And we can also start seeing this subsequently in time quite quickly in the other stages of processing along the ventral visual stream. So let's continue playing. What we see also now is that at some point, human faces become quite different from all the other objects. And we see here now in IT, a cluster of animate objects, which disappears again. At some point, it will appear in V4 later. That's what we see here. And we don't see that so much in the early visual regions. Okay, so we can also look at this representation. I'll just let it play. But we can also look at this again using these MDS plots. So let's do that. They're now shown here. So these again are MDS plots where now the images are color-coded according to their category. And there's also plots where we see the actual images. It's a little bit small, so I would suggest to focus on these colored dots here, specifically human faces, which are dark red. So you can see quite clearly when I start at the beginning again that initially there isn't much structure, but then we start to see again that there are these clusters, especially human faces. You can see them, right? And then at some point they become different from the other objects. So now they move away. And we can also see again in human IT here, warmer and cooler colors reflecting animate and inanimate groupings. So this is the data that we want to explain. So how did we go about explaining these data? What we did is we used two types of models, two classes, I would say. So one is Visuo-semantic models, and these models consist of labels for the objects that were generated by human observers. And these are both categories at different levels of abstraction and features, including object parts. And these models were quite common already for multiple decades for explaining representations in higher level visual regions. And here what's quite interesting is that, the human observers are basically providing us with labels after they've already done all the computations, right? So this model is not computationally explicit, but we know it is a good model for explaining variance. So then the other class of models is deep neural networks. So we used core nets, which are specifically built to have a similar architecture to the prime and ventral visual system. And we use two versions there. So one is a feed-forward only deep neural network, and the other is locally recurrent. And we put these together to be one class of models. So what we did next is to say, let's linearly combine the different categories and features. So that's for the Visuo-semantic models, or the different layers and time points, that's for the deep neural networks, to best explain the human MEG data at each time point after stimulus onset. So when we do that, we get the following results. So let's first look at early visual cortex. And what I'm showing here is unique variance explained on the y-axis. So I'm not showing the overlapping variance explained by the two types of models. And then time here on the x-axis. And in blue, I'm going to show you the variance explained by the Visuo-semantic model. And in green, the variance explained by the deep neural networks. So I'm also indicating when the variance explained is significantly above baseline. And what we see here quite clearly is that for early visual cortex, these deep neural networks perform better at explaining variance across most points in time after stimulus onset. But we clearly see that there is the strongest advantage early in the response, about 100 125 milliseconds after stimulus onset. That's the window where we see the clearest advantage of the DNN. Now, if we look at intermediate visual cortex, we again see that the DNNs do perform better in this early time window, but then the Visuo-semantic model also has several peaks, and they are at a later point in time after stimulus onset. And then here for high-level visual cortex, we see quite clearly that it's the Visuo-semantic model that explains more variance than the DNN, and there isn't really a significant amount of unique variance explained by the DNN in this higher level stage of visual processing. So what we see here is that there is this reversal between the deep neural networks and the Visuo-semantic models if we look at which model best explains the data once we move up the ventral visual system. So DNNs do better early in terms of the early processing stages spatially, right? And then Visuo-semantic models do better later. And we also see that the advantage of the deep neural networks is predominantly in an early time window. So this suggests that Visuo-semantic models and DNNs explain complementary representational variance. What we also did is we looked then at IT and we asked which of these components of the Visuo-semantic model contribute to the unique variance that the model as a whole explains at the level of IT. And we found that it's mostly basic level categories, such as dog, and object parts, such as ear, that explain the unique variance in IT. So to summarize these findings, here what I'm plotting now is a difference score. So I'm showing positive deflections is where the deep neural networks explain more variance than the Visuo-semantic model. And then negative deflections is the opposite. So if now first we show early visual cortex, we indeed see that in this time window, basically after stimulus onset till about 125 milliseconds after stimulus onset, you can see that the deep neural network clearly explains more variance than the Visuo-semantic model. And this time window is generally thought to reflect most predominantly feed-forward and locally recurrent processing. And that's also thinking about the architectures of the core nets that we used, that's also architecturally what they are able to do. So now if we look at intermediate visual cortex, we see again that there is this early advantage, but then it switches, and then the Visuo-semantic model explains more variance later in time after stimulus onset. So for IT, we see that it's predominantly or only the Visuo-semantic model explaining always more variance than the deep neural networks. And we also see that there is this peak that is located at about 120 milliseconds after stimulus onset. And now what we did, and this is just a visual observation at this moment, is we also included time windows from a previous analysis performed by Kiesman and colleagues that were identified to predominantly reflect forward processing from V4 to IT. And as you can see, that happens before we see the peak in IT of the Visuo-semantic model. So now what we can do as well is to identify this peak in V4 for the Visuo-semantic model. That happens later, around 160 milliseconds or so after stimulus onset. And there's also, what's interesting is that the window of backward processing, so top-down feedback, flow of information from IT to V4, so this was determined using Granger causality analysis, actually happens before we see this peak in V4. So that's quite interesting, and of course it's all very suggestive, but it does seem to suggest that first we have this forward flow of information from V4 to IT, then there's a peak of Visuo-semantic information in IT, then we have a backwards flow from IT to V4, and that's when we see the peak of Visuo-semantic information in V4. So this is, again, very speculative, but it seems to at least be consistent with this idea that there may be Visuo-semantic feedback from IT to V4 at a later time window, around 140 milliseconds after stimulus onset. And we're currently implementing further analyses to confirm this hypothesis. Okay, so let's get back to our question of what is missing here from the deep neural networks. So this work shows, first of all, that we do need to include top-down feedback. So that's an architectural change. But also, in addition to that, we do see that the deep neural networks do not fully capture or haven't fully explicitly learned these visuo-semantic features that are represented in high-level visual cortex. Now, the question is, of course, how can we change this and make them more human-like? So one way in which this may indeed be improved is to provide the networks with a more human-like learning experience. Because if we think about it, the networks have a quite impoverished experience compared to humans. And one difference that I want to focus on first is that there are clearly differences in learning objectives. So when a network is learning to categorize and recognize objects, usually it's trained in a supervised way. So it's just provided with an input and it needs to learn a mapping to a desired output, which is standardly a category label. While humans, they may be learning this way too. It's like a parent pointing to an object and saying this is a dog, for example, or this is a tomato. They, of course, are also learning in other ways, including unsupervised learning, where you can think of this more as discovering structure in the input. So one way in which this can be also implemented for neural networks is to train the networks to represent these inputs in this lower dimensional embedding space where similar inputs are grouped together and different inputs are pushed apart. So for example, showing that schematically here. And now one of the things in the lab that we're doing is to train these networks with different learning objectives to characterize what happens to the representation for these different learning objectives. So I just wanted to show you some recent work. This is not published yet, but here we're just training ResNet 50 in different ways. So what we see here is the following. In the middle, we see human IT. So this is the representation again from fMRI that we saw before. And now on the left, we see a representation of the same object images again, but now in ResNet 50 from a deep layer, layer 4, which is similar in its location in the cortical hierarchy or the visual hierarchy as human IT. So what we are going to show is how this representation evolves over training. So you'll see here epoch numbers. Sorry, I'm trying to show my mouse. Oh, over here. So this is here in the right bottom corner. And that's going to develop because we trained the network for 200 epochs. And then what we'll see is how the representation evolves over training. And that's just standard category supervision using ImageNet. And then here for ResNet 50, we also, same network, but now we train it with unsupervised learning. So we used unsupervised contrastive learning. This is momentum contrast. And again, we can see how the representation of these object images that we also presented to humans develops over time as the network is being trained. So again, we'll see movies. Now it's not dynamics over time after stimulus onset, but it's dynamics over training. So let me play this. Let's see. Oh, yeah. And as it's playing, what I'll also do is I'll take these screenshots at different moments in time. So training epoch is here on the x-axis, and on the y-axis, there's going to be supervised and unsupervised snapshots of the RDM movies. But I would focus first on the movies as I'm playing them. Yeah, so here we go. And what we can see is that there's quite early on different representations in the networks trained with these different learning objectives. So in human IT, we can see this cluster of faces, not just human faces, but also animal faces and pairs of human and animal faces. And we also see this in the unsupervised network. We don't really see that in the supervised network. And that's important to notice, right? So the unsupervised network can somehow extract information that clusters faces Now, if we look at this animate and inanimate, these clusters, right, in the human IT data, we don't really see these in the networks, as you can see, right? So that's something they're both still missing. So what we can infer here is that the way that the networks are trained definitely clearly affects the way they represent objects. And the supervised network is in some ways constrained more than the unsupervised network because the supervised network is constrained by the category labels that we provided. And that may be different labels, for example, than those categories that are ecologically relevant to humans, like faces or animate, inanimate. What we also see is that in the unsupervised network, we do see this emerge, and that emerges, in some ways, it seems to emerge more slowly, but then stabilizes also after a certain amount of training epochs, maybe around 40 epochs. There's a relatively stable representation now. So most of the changes do happen early in the training. And that's also the case for the performance of the networks and object categorization, although that keeps improving over training. I should also mention that if we now compute correlations between the human IT RDM and the network RDMs, we do see that we still do not reach the noise ceiling. So that should be mentioned, right? We're not there yet. And especially this clear indication that animacy isn't learned yet by the networks in either training regime suggests that we may need to make more changes. So I think that one of the changes that is important as well, and I'm going to show that here, is to not only look at learning objectives, but also at the input and create a more human-like visual diet. So that's something that we're currently working on. So for example, here you could imagine training on movies. And if you think about animacy, that may be quite an important source of information because animates move in different ways than inanimates. So that's something that is certainly important in terms of combining the learning objectives and the visual diet to make both of them more human-like. And then the hope is, of course, that these are in a way additional constraints. And if we impose these, that we may indeed be able to build networks that have a more human-like representation of the object images. So yeah, with that, I'm actually at the end already of my presentation. So I wanted to thank my collaborators and also my funders. And then I did want to mention that we also have postdoc positions available in the lab. So if you're interested, here is the lab website. You can check it out for more details. Thank you.
All right. Thank you very much, Marieke. This was a lovely talk. It drew a lot of questions, which we'll jump into right away. So we have a few minutes here before the break. So there's a first question here by Ayuno, and she asks, in the video you showed, was there a top-down dissimilarity signal that appears earlier in the V4 IT area and then appears in the V1, V4? So that's kind of a technical point. She might have picked up on something here.
So I think if I understand correctly, this is asking for one step further down and say, is V4 sending feedback to V1?
Right.
And I did start looking at this, but there the pattern was a little less clear, which may have to do also with the type of feedback that's being sent. So certainly if you look in the MEG data, you do see there are time windows where if you use Granger causality analysis and just ask, can the past of V4 predicts the future of V1 better than the past of V1, basically, or right, in explaining, then you do see time windows. They are a bit earlier, but they weren't necessarily aligned in our scenario with feedback, feedback thinking it would maybe be visuo-semantic because that may not be exactly what V1 is sending down. So that's a bit of an open question at the moment, and that's certainly interesting. And in that sense, we're limited in terms of the models we use by, we can just connect visuo-semantic information to feed forward or feedback, right? And that's what we can do, which doesn't mean that there is no feedback. Of course, there is, from V4 to IT. but it may be of a different nature.
Great. Actually, there's another question that dovetails really well with this one. So I'll read it from Ezekiel Williams. And the question goes, given that variance explain peaks in IT before the apparent feedback to V4, one might speculate that the feedback is not important for object recognition. Would you agree with this? And if so, what would you consider the role of this feedback to be?
Yeah, that's a good question. So, of course, I cannot say. So, first of all, it's quite speculative, the statement I'm making here about, visuo-semantic feedback. But we're doing some additional analyses to test whether, for example, you can remove information from your target region and then see what happens to the feedback if it's still if it's still there. Because if it's visuo-semantic and you take out visuo-semantic variance, let's say, from IT, then you shouldn't see that feedback anymore if you do the same analyses then to determine the window of feedback. So that's something we are doing. But then the second question is, of course, is this relevant, right? Is the feedback important for object recognition? And then you would have to do more extensive, let's say, manipulations of these visual conditions that the objects are viewed under, I would say, because there is previous research, for example, from Gabriel Kreiman's lab and others as well, Jim Ricardo's lab, showing that for example, if you introduce object occlusion, this may be a scenario in which feedback is essential, and that can be tested by having a feed-forward and a recurrent network doing that task of recognizing objects under occlusion, and then you can see that the network needs recurrent connections to be able to do the task almost as good as humans. It still doesn't exactly get there, but better than a feed-forward network alone. So in that sense, you can start to create these networks, and at the level of the network, you can lesion the feedback connections, for example, see what it does in terms of the task. So behavior is very important, and that is something I cannot address here because this was just very clearly visible images. My expectation is that feedback is still important, but if you really want to test this, you probably need to create these visually more challenging conditions and then just ask behaviorally, can the network do the task with or without the feedback connections or recurrent processing locally?
Makes sense. So I know we're almost out of time. Maybe one last quick question from Adrian Fairhall. How do you interpret the interesting temporal dynamics of the variance explained in the different areas?
Yeah, this is still somewhat of a, I don't think I can fully understand all the temporal dynamics there. What does make sense is that there is this relatively early time window within 125 milliseconds after stimulus onset that we do see variance explained predominantly by the deep neural networks in early and intermediate visual regions. And that does make sense in terms of how the networks were trained. So they were trained to do the entire transformation from image to meaning. So they also have to model these early steps, while the visuo-semantic models do not exactly do that. It's just humans giving us their interpretation of the stimulus after they have done the whole process, basically. So it makes sense. And that is really more feed-forward and locally recurrent, which matches up very well with the architecture of the networks. They don't have feedback connections. So they explain this more in the beginning. Then in terms of different peaks, that may be related indeed to feedback coming in. That's sort of the speculation I'm now making, right? But then there's other components where, for example, there's more variance explained in V1. It goes first up and then down and then up a bit again. There is this part around 200 milliseconds after stimulus onset where neither of the models does that well. And I'm not sure why. This is an open question.
Super interesting. Okay, in the interest of time, I think we'll leave it to that. Thank you once again for the lovely presentation and the great questions and engagement from everybody here. So we will take a short break, and we'll be back in 8 minutes at 3:30 Eastern Time for a keynote of this afternoon with Dr. Surya Ganguly. So once again, thank you very much, Dr. Mur, and we'll see everybody in just a little bit.
Bye.